Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad Integrated MSc PhD Entrance Examination for the subject Biotechnology conducted in the year 2021. I will be discussing only the biology questions. So coming to the first question. Which phase of bacterial growth is considered suitable to induce the expression of recombinant protein in growing culture? So it is the log phase. Answer is option B. Then put the following words from human taxonomy in correct order. So first one is primates, because that is order primates. So 4. Then comes order, then suborder, that is Haplorhini, that is 5. So 4, 5. Then comes Hominidae, that is 1. Hominidae is family. Then comes subfamily Homininae, 3. And then comes the genus Homo. So the answer here is option C, that is 4, 5, 1, 3 and 2. Then next one. Which atmospheric layer contains the highest density of gases? So it is troposphere. Answer is option D. Then amino acids can be detected by using ninhydrin. Answer is option C. Then which of the following point mutations in a DNA sequence can be identified as transition? So transition, a point mutation that changes a purine nucleotide to another purine or a pyrimidine nucleotide to another pyrimidine. So A and G are purine here, and G getting converted to A is a transition. So answer is option C. Then arrange the following in order of permeability across lipid bilayer. So carbon dioxide will be the most permeable. So 3. Then come and the least permeable will be Na positive ion. So 3 must be in the beginning and 4 must be at the end. So that is seen in the case of option C. So C will be the answer. But we will see for the others also. So 3 is greater than 1, that is glycerol, is greater than tryptophan and is greater than glucose, and that is greater than Na+. So answer is option C. The genome of coronavirus is single-stranded RNA. Answer is option A. Then match the following. Plasmodium gallinaceum infects. So it infects avian, that is birds. 1, 4. Then Plasmodium vivax infects. So it infects humans. Plasmodium berghei infects rodents. And Plasmodium novelsii infects. It infects primates. So the answer here is option B, that is 1, 4, 3, 2, 3, 1 and 4, 2. Then during the evolution, the photosynthetic organism responsible for increasing atmospheric oxygen from less than 1% to about 20% is cyanobacteria. Answer is option D. Which of the following statement is or are true? Type 1 error is the rejection of true null hypothesis. That is correct. Then type 2 error is the acceptance of false null hypothesis. Yeah, that is also correct. So 1 and 2 are correct. Okay, we'll see 3. Type 1 error is the rejection of true alternative hypothesis. No. 4. Type 2 error is acceptance of false alternative hypothesis. So 3 and 4 are wrong. Only 1 and 2 are correct. So answer is option C. Then which of the following is an important mechanism of bacterial transformation? So it is conjugation. Answer is option. Which one is the laughing gas? So it is nitrous oxide, N2O. Answer is option D. Which is the correct order of the following steps in southern hybridization? Binding of primary antibody, transfer onto membrane, denaturation of proteins, reduction of hydrogen peroxide. So the first step will be denaturation of protein. So D3. Then transfer onto membrane. So 2. Then binding of primary antibody and reduction of hydrogen peroxide. So the answer will be option D, that is 3, 2, 1 and 4. Then parental alleles can be detected by using the technique. So they can be detected by using restriction fragment length polymorphism or RFLP. Then activation energy of a chemical reaction can be evaluated using Arrhenius equation. So this is the Arrhenius equation. This is the Arrhenius equation. K is equal to A e raised to minus EA by RT, where K is the rate constant, A is the pre-exponential factor, E is the base of natural logarithms, and EA is the activation energy, R is the gas constant, and T is the temperature in Kelvin. So this can be, that is, activation energy of a chemical reaction can be evaluated using Arrhenius equation. The next one. Which of the following is a part of forward genetic approach? Now I have already discussed what is forward genetics and reverse genetics in one of my previous question papers. I think you will get it in University of Hyderabad MSc Plant Biology and Biotechnology question paper for the year 2021. Anyway, I will tell what is reverse genetics. It is an approach to understand the function of a gene. So what you do is, first you select the gene, then you generate a null mutant of the gene, then you analyze the phenotype. So like that you will analyze the function of a gene in the case of reverse genetics. Whereas in forward genetics, it is an approach to determine the genetic basis for a particular phenotype. So you will start from the phenotype, you will screen all the collection of mutant phenotypes, then identify the gene causing the phenotype. That is in the case of forward genetics. So which of the following is a part of forward genetic approach? So it is option A, that is generation of random mutation in the genome that confers a specific phenotype. That is the case of forward genetic approach. So answer is option A. Then a protein has 6 cysteine residues. In how many ways 3 disulfide linkages can be formed? So these are the number of ways in which 3 disulfide linkages can be formed. C1 is the first cysteine, C2 is the second, C3 is the third cysteine, C4 is the fourth cysteine, and C5 is the fifth cysteine, and C6 is the sixth cysteine. So cysteine double bond, that is disulfide linkages. So with C1, C2 and C3 it can form disulfide linkages. So like that it can form 15. So the answer is option C, that is 50. The next one. Which of the following is the characteristic of cancer cell? Density dependent inhibition, contact inhibition, loss of anchorage dependence. That is characteristic of cancer cell. So answer is option C. Then restriction and recognition sequence. That is 5 prime. 5 prime CTG CAG 3 prime. Restriction enzyme B has recognition sequence 5 prime GC. Based on this inference, you can infer that. So now enzyme A, it has got how many nucleotides? 1, 2, 6 nucleotides. So 4 raised to 6. That is, enzyme A should cut once every 4 raised to 6 base pair of the genome. So what is 4 raised to 6? So 4 raised to 6 will be 4096. 4096. So enzyme A will cut once every 4096 base pairs in the genome. Now how much is 4 raised to 4? So 4 raised to 4 will be 256. That is in the case of enzyme B. So after every 256 base pair it will be cutting. Whereas enzyme A will cut only after every 4096 base pairs. So our answer here is option B. That is, enzyme A will cut the genome into fewer pieces than will enzyme B. Why? Because enzyme A will cut only after every 4096 base pairs, whereas enzyme B, it will cut after every 256 base pair. So the number of fragments generated by cutting with enzyme B will be more than the number of fragments you get by cutting with enzyme A. So enzyme A will cut the genome into fewer pieces than by will enzyme B is the correct answer. So the answer is option B. Then, which of the following conditions lead to maximal expression of the lac operon? So, lactose is present, glucose is absent. Yes, because E. coli prefers glucose over lactose. So, when glucose is present, it will not synthesize the lactose metabolizing enzymes. So, when lactose is present and glucose is absent, it will have no choice but to utilize lactose, and this lead to the expression of lactose utilizing or metabolizing enzymes of the lac operon. So the answer is option A, lactose present and glucose absent. Then arrange in the sequential order the process of Western blotting. So incubation with secondary antibody, run the protein samples on SDS gel. Then incubation with primary antibody, transfer to nitrocellulose membrane, blocking. So first you have to run the protein samples on SDS gel. Then after that you have to transfer them to nitrocellulose membrane. Then you have to block them. Then you have to treat them with primary antibody, that is incubation with primary antibody, and then finally with incubation with secondary antibody. So the answer is option D. Then match the correct pairs. Amino acid, it is LEVO. 1 is 5. It is in a natural form, it is the L amino acid that is present. Then a deoxyribose, it is dextro. So 2, 4. Then SN1 product, it is racemic mixture, and SN2 product, it is inversion. So the answer here is option C, that is 5, 1, 4, 2, 4, 2, 1, 5, 2, 4, 3, 1 and 4, 3. The next one, the site of hematopoiesis during the 5th month of an infant is, it is in the liver and the spleen. Answer is option. Then identify the correct statement. Methylation of DNA helps in gene silencing. That is correct. Then regulation of promoter activity is the only way to regulate gene expression. So eukaryotic gene expression, it can be regulated during transcription and RNA processing which take place in the nucleus, as well as during protein translation which take place in the cytoplasm. So that is not the correct statement. Then 3' UTRs often contain the binding site for miRNA. Yes, miRNA induce mRNA degradation and translational repression by binding to the 3' UTR of the mRNA. So answer is option 3 is also correct. Then miRNA enhance the translation process for a particular mRNA. It doesn't enhance the translation process, it will degrade the mRNA. So 4th is also not correct. So 1 and 3 are only correct. So answer is option A. Then the scientist who discovered the process of phagocytosis, it is Eli McIntosh. Answer is option C. Then match the correct pairs for the functions performed by plants. Phytosiderophores. So it is chelate and solubilize Fe3 plus to make it available for uptake. So C3. Then phytoextraction, it is removal of toxins from soil. Then phytostabilization, it is complexation and immobilization of toxins within the soil. Phytodegradation, it is removal of organic contaminants in the rhizosphere. So answer is option C, that is 1, 3, 2, 4, 3, 2 and 4, 1. Then the defense mechanism presented by human skin is, it is innate immunity. Answer is option. Then the shape of the tobacco mosaic virus, it is helical. Answer is option C. Which genetic elements are covalently closed, circular, self-replicating and transmissible? So they are none other than plasmids. Answer is option C. Which of the immune cells are produced in bone marrow but mature in the thymus? It is the T cells. Answer is option B. The Sanger sequencing method is based on dideoxy chain termination. Answer is option A. This is a repeated question. Identify the correct statement related to cytoplasmic male sterility in plants. It is caused by mutations in the mitochondria. That is correct. Then caused by mutations in the nucleus, no. Mitochondria are inherited from the cytoplasm of egg cell. That is also correct. Then follow Mendel's law of segregation. No, they are actually maternal inheritance. So 1 and 3 are correct. Answer is option 1. Which of the following regarding Ames test is true? So this question was deleted. Okay, what is Ames test? It is used as an initial screen to determine the mutagenic potential of new chemicals and drugs. For that only Ames test is used. Okay, now the question is deleted, so I am not discussing much into it. Then next one, match the correct pairs. So phosphofructokinase. Phosphofructokinase is allosteric interaction. So 1, 3. Then glycogen synthase. It is covalent modification. Beta galactosidase. It is control of enzyme synthesis, and lactate dehydrogenase. It is product of inhibition. So answer is option B, that is 1, 3, 2, 2, 3, 4 and 4, 1. Then cyclin is associated with, it is associated with mitosis. Answer is D. The scientists who are involved in initial experiments of phototropism and geotropism. So they are Charles Darwin and Theophil Selinsky. Answer is option D, that is 2 and 4. Then what is the main epigenetic event in the genome? So it is methylation of DNA and histone modification. The first step in allopatric speciation is, so allopatric speciation occurs when biological population become geographically isolated from each other to an extent that prevents and interferes with the gene flow. So answer here is splitting of a population into 2 or more groups by geographic barrier. So answer is option A. Then the amino acid which accumulate in plant during freezing tolerance, it is proline. During stress condition, it is amino acid proline that is present maximum. In next one, the enzyme which convert phosphoenol pyruvate to malate, it is PEP carboxylase and malate dehydrogenase. Answer is option A. Then match the correct pair of alkaloid with their plant source. So ajmalene. Ajmalene is Rauwolfia serpentine. Then codeine is Papaver somniferum. Scopolamine is Hoescamus niger, and quinine is Cinchona officinalis. So the answer is option D. That is 1, 3, 2, 1, 3, 4 and 4, 2. Then in which of the following cells lysosomes are absent? So lysosomes are absent in the case of erythrocytes. Answer is B. Then which of the following pair match with each other? Polymerase involved in transcription of ribosomal RNA except 5S rRNA. So it is RNA polymerase 1. So 1, 4. Then polymerase that incorporates wrong bases in DNA and is used in translation synthesis. So that is DNA polymerase 5. Then you have polymerase that is involved in transcription of tRNA. So it is RNA polymerase 3. And polymerase that makes DNA from RNA template and remains associated with the enzyme, it is telomerase. Answer is 4. So the correct answer is 4, 5, 3, 2. Sorry, 1, 4, 2, 5, 3, 3 and 4, 2. Answer is option C. Then the two reactions of two enzyme A and enzyme B are studied at pH 7. Both the enzyme produce lactose and have the same Vmax. Enzyme A has Km of 2 millimolar while enzyme B has Km of 5 millimolar. Consider the following statements when enzyme A and B both carry out their reaction at pH 7. So Km is inversely proportional to the affinity of the enzyme for its substrate. So enzyme A, it is having a Km of 2 molar. So that is lesser Km value, higher will be the affinity for the enzyme. So enzyme A will produce more lactose than enzyme B when substrate concentration is 5 millimolar. So the answer here is option A. 1 is only correct. So A 1 only is correct. Then what is the characteristic of protein quaternary structure? The quaternary structure of a multimeric protein always depends upon the primary structure of the subunits. Answer is option D. Then 67, this question was deleted. Then which of the following pair match with each other? Degrades mRNA from RNA DNA hybrid. So it is RNase H. So 1 4. Then separates the two daughter chromosomes after replication. So it is topoisomerase 3 2. Cleaves the phosphodiester bond of RNA. So it is RNase A, that is 3 5. And unwinds the double stranded RNA, it is helicase. Okay. Answer is option 1 is 4, 2 is 3, 3 is 5 and 4 is 2. Answer is option B. Then match the given microbial products with their producing microorganism. So amino acid, one will be Corynebacterium glutamicum, 4. Then organic acids will be Aspergillus niger, that is citric acid can be produced, that are organic acids. Then 3, that is vitamins. Vitamins by Erythrothecium ashby, 3 2. And polysaccharides by Xanthomonas. So answer here is option D, that is 1 4, 2 3, 3 2 and 4 1. Then trypsin and chymotrypsin are example of digestive enzymes, and the active site of these enzymes consists of the catalytic triad. Which of the following amino acid residues are involved in the active site? So they are serine, histidine and aspartate. So the answer here is option C. So that is it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.